start. Good afternoon, everybody. Today's class for practical class is on cestodes and trematodes. So, what are cestodes and trematodes? They are basically helminths. Helminths are classified into two phyla, platyhelminths and nematohelminths. So, what do you mean by platyhelminths? Platy means flat. So, both these cestodes and trematodes, they come under platyhelminths, and these are flat worms, basically. And their body is divided into head, neck, and the body. So how do we differentiate and with cestodes, trematodes, and nematodes? As I said, cestodes and trematodes, they come under platyhelminths, and nematodes are nematohelminths. So, cestodes and trematodes are flat worms. So, cestodes are tape-like and they are segmented, while trematodes are leaf-like and they are unsegmented. The body is unsegmented. But classically different are the nematodes, whose body is cylindrical and they are unsegmented. So in cestodes, their sexes are not separate; they are monoecious, and trematodes are also monoecious except for schistosomes, and nematodes are dioecious. That means their sexes are separate. So as I said, the body has head and neck and body. So the head end of cestodes have suckers and hooks, and in trematodes, the head end has only suckers, but hooks are absent. In nematodes, the head end has neither suckers nor hooks. And there is no elementary cavity in cestodes, and it is incomplete in trematodes, but it is completely developed in nematodes. The body cavity also is absent in cestodes, trematodes, and but present in nematodes. So, the main organisms that come under cestodes are taenia and various other organisms, but taenia is most important. Under taenia, there are two species, saginata and solium. Taenia saginata is called as beef tapeworm, and Taenia solium is called as pork tapeworm. They're common names. So, how do we differentiate these two worms, adult worms? Tinea saginata is usually very lengthy, of around 5 to 10 meters, and Tinea solium is 2 to 3 meters long only. The head end, which is also called as scolex, it is large, quadrate, but without rostellum and hooks in saginata. But in solium, the scolex is very small. It is globular with rostellum and hooklets. Hooks. So the proglottids, the body has various segments called proglottids. Uh, these proglottids, based on their maturity levels, they are classified as immature, mature, and gravid proglottids. So based on the proglottids structure, they can be differentiated. So in saginata, the number of proglottids are higher than solium, which are around 1000 to 2000 in saginata and less than 1000 in solium. Also, the way of expulsion is different in both the organisms. Like in saginata, they are expelled singly and may force the anal sphincter. But in solium, they are passed out. They are passed out passively in chains of 5 to 6. And also, they can be differentiated based on the structure of uterus. Where in saginata, the uterus has lateral branches, 15 to 30 on each side, and thin and dichotomous. But in solium, uh, there are lateral branches which are 5 to 10 on each side, lesser than that seen in saginata, and they are thick and dendritic. So this is the head end, that is colex of both the species, saginata and solium. Here in saginata, as you can see in this diagram, there are four suckers. These four suckers, but hooks are not seen, hooks or hooklets. But in solium, you can clearly see the hooklets present at the topmost end and along with the four suckers. And also there are double row of hooks. So how do we differentiate these gravid segments of Tinea saginata and Tinea solium? So in the proglottids, as I said, are classified into immature, mature, and gravid. Mature proglottids have uterus developed quite well developed, and then gravid segments, the uterus is filled completely with the eggs. So, based on the gravid proglottid structure, they can be differentiated. In solium, the length and breadth of the gravid segment is around 12 mm by 6 mm, but in saginata, it is longer. The length is 3 to 4 times than its breadth. And also by the way of expulsion, as it was said earlier, in solium they are expelled passively in chains of 5 to 6 at a time. And in saginata, they are expelled singly and may force the anal sphincter. So as seen in this diagram, this is the internal structure microscopic view of gravid segment. Now, this is the gross specimen of Tinea worm. Here, if you are able to appreciate, this is the Tinea worm with various segmented, which are, you see closely, they can be divided class. They can be seen like immature, mature, and gravid segments here. But when you come to your practical class in person, these can be shown. Then you will be able to appreciate. But here, if you see, you can appreciate the different segments based on their length and breadth. Okay. So this is the tapeworm. And this is the gross specimen of just the proglottids, not the entire worm, but just the proglottids, which can be appreciated. दिख रहा है क्या? So now coming to Cysticercus cellulosae, this is the larval form of Taenia solium. So, and the disease caused by this larval form is cysticercosis. So, how do man acquire the infection of cysticercosis? Cysticercus cellulosae. So, it occurs by ingestion of contaminated food like uncooked vegetables, which are infected with eggs, and also drinking contaminated water. And there is also possibility of auto-infection from adult worm by reverse peristalsis or unclean personal habits. Okay. This larval form can be seen in many parts of the body. Mostly it occupies the subcutaneous tissue, muscles, and maybe brain and eye sometimes. But mostly it is seen in subcutaneous tissue and muscles. So it is the most common cause of adult epilepsy, where symptoms vary depending on the site of involvement of this larva. So usually it tends to calcify and becomes obsolete by 5 to 6 years. So the diagnosis of this cysticercosis and the presence of Cysticercus cellulosae larva is usually by radiology or histopathology, as can be seen in this diagram, and the treatment is surgical excision. Now coming to Diphyllobothrium latum. This is called fish tapeworm. It is endemic in areas where humans frequently consume raw or pickled fish. So most persons with diphyllobothriasis are asymptomatic, and when patient presents with symptoms, they can present with abdominal pain or indigestion or dyspepsia and passage of proglottids. So gross examination of stool is important in these cases, and it can be diagnosed by identifying the proglottids present in stool or characteristic eggs that are passed down into the feces. So this is the diagram of egg of Diphyllobothrium latum. Now coming to Hymenelepis nana, H. nana, which is also called as dwarf tapeworm. So this tapeworm, it infects the small intestine. Its habitat is small intestine of man, and it has head, neck, and body. As I said, the neck is very long in Hymenelepis nana. So the entire worm measures around 15 to 14 mm, and the head has rostellum with hooks at the topmost end, and below that it has 4 suckers, and these hooklets, there are a single row of hooklets which are around 20 to 30, and in the rostellum part. And the proglottids, there are marginal genital pores on same side of the proglottids, and the transverse sac like uterus with lobulated walls is present. And there is also an testis, 3 testes are present in each proglottid. So how do you identify the eggs of Hymenelepis nana? So this is the diagram, photograph of Hymenelepis nana egg, which is non-biostine and round to oval. It has two distinct membranes, the outer membrane and the inner membrane. This is the embryophore, which is embryophore or oncosphere, which is 6 hooks, and there are also 4 to 8 polar filaments spread out between these two membranes. Here, there can be polar filaments, and this is all the eggs of both H. nana, Hymenopus nana, and Taenia are almost similar, which makes them very difficult to differentiate. But the only feature which can help in differentiating both of them is that the eggs of Taenia are biostine and Hymenopus nana are non-biostine. Otherwise, both of them look almost similar. So these eggs are spherical and bistained in Taenia. H. nana is spherical, non-bistained. The inner embryophore is brown and thin-walled, which has radial striations. These radial striations are not seen in Hymenopus nana egg. And it contains an oncosphere with 3 pairs of hooklets, Taenia, and similar in Hymenopus nana also. And Taenia eggs do not float in saturated salt solution. And Hymenopus nana, they float in saturated salt solution. And here in H. nana, there are 4 to 8 polar filaments in between the outer and inner membrane. Now coming to flukes, which are trematodes. These trematodes are leaf-like organisms. So Fasciola hepatica, Fasciolopsis viscae, Paronimus westemani, these are the organisms which come under trematodes. So I'll be dealing with Fasciola hepatica and Fasciolopsis viscae. So Fasciola hepatica, so the adult worm, as seen in this picture, it is large, leaf-shaped, 3 centimeters, 3 by 5, 3 centimeters in length and 1.5 centimeters in its breadth. It has 2 suckers. Anterior end is with oral sucker it has a conical projection in the anterior end, which has oral sucker, and it forms the anterior end as it is connected to the entire body. It forms to shoulder kind of structure anteriorly. So this the posterior end is rounded here, and there are internally the intestinal theca. They bear lateral compound branches. Coming to eggs are large, ovoid, and operculated. As you can appreciate in this diagram, the operculum, the lid-like structure that is present. So, and these are biostane. They do not float in saturated salt solution, and it contains unsegmented ovum inside this egg. The definitive host of Fasciola hepatica are sheep, cattle, and man, and the intermediate host is limnae species, which are snails. Now coming to adult Fasciolopsis viscae, here it is called as giant intestinal fluke. Here you can see the anterior end is not so conical, which can be differentiated from Fasciola hepatica, which is really very conical and forms 2 shoulders with the body. Here that is not seen. It is not completely conical, and you won't be able to appreciate the shoulders type of structure also here. And this lives in small intestine of man and pig. And the mode of infection is by eating infected plants as raw foodstuff. The infective stage is metacercaria, which is infected cercaria. See, here for all the organisms, you should be able to know the entire life cycle of all the organisms. Only then you'll be able to understand how it is infecting man and in what stage it is infecting, which stage is infective, and how every stage of the organism is important. So here the infective stages, metacercariae, which are ancestor cercariae. So this is the largest trematode which infects man, uh, whose length is around 20 to 75 mm and 8 to 20 mm in breadth, and its thickness is around 5.5 to 3 mm. It is elongated, oval in shape. Like unlike Fasciola hepatica, it does not possess any cephalic cone, as I said earlier, and two intestinal cecae don't possess any lateral branches. So here there are no lateral branches, and in hepatica there are lateral compound branches. And also here there is the conical anterior end with shoulders, and here there is no conical end with shoulders. So this is the gross specimen of Flukes. Here in this specimen bottle, both the facial oxydosta and facial hepatica both are present in this. Here you can appreciate the anterior end and the posterior end. Okay, the leaf-like structures which are very flat worms. Okay. Now Dr. Monica will explain Paragonymous vestment. Good afternoon. I will be talking about Paragonymous vestimani. So this Paragonymous vestimani, they are also known as Japanese lung fluke or Oriental lung fluke. So almost 30 species of genus Paragonymous is known, which can infect both human and animal, in which only 10 species are known to cause infection in human. In this, most common is the Paragonymous westernmani. So this Paragonymous westernmani, they are mostly found in a specific geographical regions such as in Eastern Asia or in Southern America. In India, they are found endemic in Manipur state. So, coming to the life cycle of Paragonymous festimani. So, first the unembryonated eggs are being excreted in sputum, or if they are being swallowed, so they can be seen in stool also. In external environment, this unembryonated egg will become embryonated and this become embryo. After that, the metacidia will hatch and it will then look for its first intermediate host, that is the snail. So it will penetrate through the soft tissue of the snail and it will undergo its further development, that is from a sporocyst to radiate to cercariae. This cercariae will then invade its second intermediate host, that is the crayfish or the crab. So, in the second intermediate host, this cercariae will insist to form metacercariae, which is the infective stage. Human beings get infection by consumption of unadequately cooked crayfish or crab. After infection, this metacercariae will exist the intestinal wall and it will penetrate the intestinal wall, then it will reach to the peritoneal cavity, and from there it will reach the abdominal wall, and then to the diaphragm, and from diaphragm it can reach to lungs, and in lungs these metacercariae will then transform into the adults. So, so I've already told that there are two intermediate hosts. First is snail, and second is the crayfish or crab, and man is the definitive host. So coming to the clinical features, so it could be like acute or chronic features. In acute, there will be diarrhea, abdominal pain, fever, urticaria. While in chronic, it will mostly it will be a pulmonary manifestation such as cough, cough with expectoration, then hemoptysis, and other radiological finding you will get. So in Cough and other radiological findings that you will get. These are the clinical features. One typical thing about clinical feature is that in the sputum will be of brown color, and the brown color is not because of the blood, but it will because of it will contain the eggs of the Paragonimus, which will give it a brown color. And because of its clinical feature, that is fever, cough, weight loss, it is mostly confused with the tuberculosis infection. So in regions where they are mostly endemic, we should keep this diagnosis also in mind of Paragonimus. Then coming to the features, so this is the egg of Paragonimus westermani. It is a golden brown color, and as you can see, it is an oval, and we can see a flattened operculum in it. It measures around 80 to 50 micrometer, and it contains unsegmented ova. So after clinical features, next is the epidemiology. The epidemiology, I've already told that they are mostly found endemic in the Eastern Asia and in parts of Southern America. Then treatment is like according to CDC, the record choice is paracequintal, which is around 75 mg per kg, which is given in 3 divided doses for consecutive 3 days. So this is the slide showing the adult Paragonimus westermani. So the adult form, it will look like a coffee bean structure, brown in color, with thick fleshy muscular structure, and the length will be around 8 to 12 millimeter, and breadth will be 4 to 6 millimeter. It has strong suckers. Both there will be oral sucker as well as ventral sucker. Ventral sucker will be located slightly pre-equatorially, and it will have two intestinal cecae, which will be unbranched and which will be extended caudally. So mostly you have to keep this organism in mind when you are in an endemic region such as in Manipur and in Asia, because they are mostly confused with the tuberculosis. Thank you.